and talk about what we talked about yesterday. So, spent a lot of time, even since you guys were freshmen, talking about transcription and translation and central dogma and gene expression, blah, 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 right? This is all about how do you get from the genetic material ultimately to curly hair, right? Or brown eyes or freckle on my, or whatever, all right? So we've talked a lot about to messenger RNA. Then the messenger RNA by the ribosome brings in tRNA to then bond amino acids together to make proteins. Cool. Well, one of the things that you have ever thought about until recently is as the tRNA in, they bring in the amino acid, and the tRNAs leave, and they don't have an amino acid anymore. How do those tRNAs get another acid? Hey, there's an enzyme. What do you know? There's an enzyme called amino-tRNA synthetase, or AATS, or ATS, or ATS, or whatever you want to call it, in all honesty. And this is a cytoplasmic enzyme that takes the amino acids from your diet, from your nutrition, reattaches the uncharged tRNA to the amino acids that we eat to recharge the tRNAs, so that way the charged tRNAs can be involved in translation and the cycle continues. So here is kind of a zoomed in cycle, right? Yes.
Is the, what's it called? The
AATS?
AATS. Is it just in the cytoplasm?
Yes.
Okay.
Only in the cytoplasm. It's not in the nucleus at all.
Oh.
Cool. Interesting question. So then step back, right? And then start thinking, seeing that tested in the stomach and the small intestine, then absorbed into the blood, right? Then as the blood flows through the arteries and then the arterioles and then the capillary and then the amino acids diffuse in the cell. And then there's good old AATS, right? Bonds the uncharged tRNA to a specific amino acid. And then now here's a little bit more detail. The whole time trying to take everything that we've talked about, right? And build all this stuff together. That's the whole point, right? You take what we know about enzymes and what we know about cell transcription, translation. Oh yeah, the stomach and the intestines and the microvilli and the villi and the blood and the heart and the blood. It's just this big connected content area. It's just amazing. Cool. So let's do a fancy book diagram looking at image 1715. And I need to get my purple crayon that I was using earlier too. So the whole time that we've been talking about AATS, so I want to be sure then this, and this is AATS. AATS, AATS, and AATS. So that is the enzyme that I've been shading in purple. I'm talking about. It's a cool enzyme. Has a tremendous responsibility, very, very important role, and most people have no idea that it even exists. And then once you start talking about it and people go, oh, of course, RNAs come in and out. Where do the amino acids come from? This isn't magic, right? It's not some big mystery. This is all based on cycles, and the more biology that you learn, you start seeing how the cycles start to connect and the cycles start to overlap, and they all work. Pretty soon what you learn in this class and what you learn in this all fit together. For those who get into biology or actually any science as a major, when you first start in your major as a freshman and a sophomore, taking what you see as these disconnected classes. And then the farther you get into your major, usually by about the later part of your sophomore year and the junior year, you should start seeing these classes start to come together, and your professors start making connections, and you're like, oh, this is what, hopefully, that's the way it's presented to you. All right, so figure 17, good old tRNA synthetase. So what are we looking at? This is an amino acid. Okay, then you know about enzymes and you know about the active sites of enzymes. All right, so this enzyme happens to have a 3-part active site. Okay, AATS has an active site that requires a specific amino acid, ATP, and eventually an uncharged tRNA, a very specific tRNA, will fit right in here. So this active site actually has 3 regions. So here is your ATP, power the process. So here's your adenosine, and here's a phosphate. Now, as a biologist, be a little lazy here, and this is where the chemists complain about, you biologists, where we only write a P phosphate, and to a chemist, a P is of course phosphorus. But I'm a biologist, so I can round some corners. It's okay. All right, so here is your specific amino acid. Here is your ATP. Okay. Now the ATP is then broken down, or the fancy term is it's hydrolyzed. So here's your adenosine, and then 2 of the phosphates are removed. And so there is actually a high energy bond left right in there. Okay. IB is never going to be specific to ask you about the fact that this is AMP. All right. I'm going to mention it because you will find out that when you get into cell biology, AMP, it's called cyclic AMP. This stuff is everywhere. This will be really, really important later on when you're about 20, 21. All right. So the ATP is then broken down. All right. Then what comes in is a very, very specific tRNA. Okay, so there's 2 things that you should be tuned into. First of all, the fact that this tRNA is uncharged, right? And this anticodon right there matches the active site enzyme. It is very, very specific. Okay, so then RNA comes in. The MP, so here is your adenosine with your single phosphate. The AMP then adds, while the uncharged tRNA, I really want to accentuate, right, that the uncharged tRNA comes in, and then your amino acid here, your specific amino acid, and uncharged tRNA bonded. That's why that high energy bond with amino acid, that actually powers physical union between the the tRNA and usually a carbon on amino, and what attaches your tRNA to your amino acid. Okay, so you get your charged tRNA. Okay, so once you sit back and look at this process, it's pretty straightforward, right? You bring in, let's get rid of the details here, right? You bring in an amino acid and an ATP. You break down the ATP to build up the energy here. You then use that energy to bond a specific amino acid to the uncharged tRNA. And then you release the charged tRNA. This charged tRNA goes to the ribosome. The ribosome then takes the amino acid and uses that amino acid to synthesize protein, and then the uncharged tRNA simply then just cycles back, right? You then produce more ATP by cellular respiration, and you eat more amino acids, and the process just keeps right on going, okay? So let's label some items now that we've talked about the picture, and you will have a completed handout. How do you feel about the process?
Pretty good.
Okay. I understand that it's unfamiliar, but that doesn't make it difficult. Just because it's unfamiliar doesn't make it difficult. Okay. So then the active site, let's make that thicker. I don't like how that ink lifted right there. There we go. So the active site. The active site bonds a specific amino acid. Okay. And then the ATP is hydrolyzed. What does that mean? Well, is when bond broken. Okay, so the ATP is hydrolyzed into technically AMP, right? That stores energy in that bond right there. Okay, so then the stored energy is then used to make that bond right there. So now a specific tRNA binds to a specific amino acid. So I am a lot when I first teach this, especially freshman year. You know, the tRNA with the anticodon brings in a specific amino acid. I get a student every year that asks, hold it, how does the tRNA bring in the right amino acid? How does the tRNA know to bring the right amino acid? Well, I can't look at a 14-year-old and say, well, there's amino acyl tRNA synthetase. Oh, come on, it's way over their head. So what you instead say is there are enzymes that bond that tRNA to a specific amino acid. Ready? And we'll talk about it later. Okay, so understand that this is just another example of layers, and there's layers beyond this, and there's layers beyond that. There's always more. Okay, and then ultimately, then here, your charged tRNA is released. Now, name of the enzyme is amino acyl tRNA. And I was surprised that none of you asked, why is it amino? Because the real for that molecule right there of the RNA bonded to the amino acid is really amino ACY. It's such a weird word. TR, but nobody amino tRNA. There's no way anybody says that. We just charged tRNA. Okay. So usually when IB asks this question, they ask you the role of tRNA and the AATS enzyme, or they give you blank diagram and they ask you to fill it in. 24 years, I've never seen IB ask you to draw this diagram from scratch. Okay, I'm not saying they couldn't, but I'd be really surprised if they did. All right, they ask you to talk about it in general terms like this, okay? Or if specific, they give you the diagram and then say, tell us what's going on here. Or they just give you the enzyme and then you have to say, hey, an amino acid comes in, an ATP comes in to power the process, and an uncharged tRNA comes in. Cool?
Yep.
All right. So then I want to show you an animation of translation that will introduce a diagram because we have 2, maybe 3 questions left in Core 7, and they're all pictures. Yes, I am presenting. Look at me. All right, so here we have messenger RNA. All right, now when we draw messenger RNA, we always say that the process of translation starts. That's accurate, okay? That's where the first tRNA comes in. That doesn't mean that the AUG is right at the end of the messenger RNA. There are early bases upstream of the start codon, okay? These bases work in matching the messenger RNA to the small subunit, right? So here you have your messenger RNA, there's a start codon AUG, and then your small subunit comes in first, and that small subunit cradles the messenger RNA, okay? Then your tRNA comes in. The tRNA, of course, has the amino acid MET, which is methionine, okay? So all proteins in all living things on the surface of the earth start with AUG and methionine, okay? Then your large subunit now comes in, and that's the cap, and that locks this whole complex together, okay? Now, notice how when the ribosome shifts, the tRNA that sits here, the tRNA doesn't move. It's the ribosome that moves, okay? So here's your next tRNA. Notice the bond, and I wish it made this much more obvious. Watch that little line. Do you see that line shift over, right? The methionine is bonded to the first tRNA, then the next tRNA comes in and just, it's super subtle, right? So when that bond forms, this methionine and valine is now attached to this transfer RNA. What's attached to this first tRNA now?
Nothing.
Nothing, okay? Making this first tRNA uncharged. Okay? Now, tRNAs are sitting on the messenger RNA. They're not going anywhere, okay? And as the ribosome moves, as the ribosome translocates, all right, we say that the tRNA that's in the P site moves to the site, and that can be misleading because when we say things like tRNA moves to the E site, is the tRNA really moving?
No.
No. What's moving?
The ribosome.
The ribosome, okay? Really important for your understanding. I don't want you thinking that the tRNA move around like chess pieces on a board, okay? Once the tRNA settles down anti-codon to codon, the tRNA remains stationary, okay? And it's the ribosome that translocates. So now as this ribosome shifts to the right, this uncharged tRNA, the uncharged RNA will then end up in the E site and be ejected, okay? Then there you go, just like that. And now this tRNA that has the growing polypeptide is sitting in the P site, and now this process repeats, okay? The next tRNA comes in, we'll shift the bond from right here, the valine will bond to the next amino acid, the peptide will be transferred over to the next tRNA, and then the ribosome translates. There's your peptide bond, and then translocation, and then next tRNA, and then there's your peptide bond and translocation. While this is a simple animation, it's really effective, okay? So, does the translocation of the ribosome make sense?
Yes.
Okay, all right. So, then you have the stop codon, right? UGA, UGG, there's 3 of them. And the receptor protein then comes in stop codon, and then this whole complex scatters, disconnects, dissociates, okay? So, I strongly recommend that you go in YouTube and find translation and watch 3D images or animations of this. Watch cartoon creations of this. Watch any form of movement on process, and then after you watch it again, and then watch it again, and then watch it again. I need you to have that library of animation in your head. It'll take you about 5 to 10. Just jump on YouTube, type in translation, and seriously watch some videos, okay? Because once you start seeing these different translation, you'll start to see the patterns, and you'll start to understand that the tRNA anchors into the messenger RNA, and the RNA doesn't move. The ribosome locates, and that's what gives us the illusion that the tRNA is moving from A site to P site to E site. That's really, the tRNA isn't moving at all. Transfer RNA stays stationary, and the ribosome translocates. Okay, I think that up, do me a favor, 5 to 10 minutes on, big for you guys, right? Watch a couple videos. You tomorrow afternoon, 2 o'clock on Zoom, and we'll an hour with Miss Macias and prepare for the TF